So this is also important for essay. So PS of dicot stem or PS of sunflower stem. The primary stem of dicot plants shows this structure. So this is ground plan of PS of dicot stem. If you enlarge, if you observe it under the microscope, you can find the structure under the compound electron microscope. The TS of dicot stem shows three parts: epidermis, cortex, and stem. Epidermis is the outermost layer. Generally, in almost all the parts of the plant body, epidermis is single layer. Epidermis, this is the outermost layer, a single layered structure. Single layer structure called epidermis. Epidermis is the outermost layer. It encloses. Could you give me the sharing option? Okay. So this is epidermis.
Excuse me, sir.
First part.
ఒక్కసారి నేను యూట్యూబ్ లింక్ ఇచ్చేసి మీకు హోస్ట్ ఇచ్చేస్తాను 1 minute ప్లీజ్.
Okay. Okay. ఇవ్వండి. నేను కంటిన్యూ చేయాలమ్మా క్లాస్? BS of dicot stem shows three parts: epidermis, cortex, and stem. Epidermis is the outermost layer. If you enlarge this part, you can see this structure. This is the enlarged portion. This is also called as sector. This is epidermis. This is epidermis. Epidermis is a single layered structure. The cells are rectangular in shape, which are closely packed, generally tubular cells. And all the cells are living cells. They have vacuolated protoplasm and thin cell wall. The outer surface, this is the outer surface. The outer surface is covered with cuticle. Thin cuticle layer will be also there. Since it is aerial part, when you see the epidermis of monocot root or dicot root, you cannot see the epidermis. You cannot see the cuticle layer. Since the stem is aerial part of the plant body, it has to be protected from adverse environmental conditions, that is temperature, rain, and humidity. That's why the outer surface of stem is always covered with a thin cuticle layer. Thin cuticle layer. That is the characteristic feature of epidermis in stem and leaf. Here you can see if it is young stem, because without secondary growth, the primary stem, in primary stem, the epidermis shows stomata. You need to remember 3 points in epidermis. Epidermis is a single layered structure. It is outer surface of the epidermis is covered with cuticle layer, and you can find stomata. You can find stomata. Stomata are aerating pores, minute microscopic aerating pores. They help in exchanges of gases. These stomata are present only when the stem is young. Generally, epidermis is green in color, since it is having, in some herbs, you can find chloroplast also. But in some trees and shrubs, you cannot find chloroplast in the epidermis. This is about epidermis. I'll repeat once again. Epidermis is the outermost part of the stem. That is first point. It is single layered structure. It is made up of tabular cells which are closely packed. All the cells are living with vacuolated cytoplasm. The outer surface is enclosed with thin cuticle. You can find few stomata. This is about epidermis. Next, cortex. This is the second part. This is the part of the stem present between epidermis and stele. This is the stele. This is the stele. If you want to see the stele, see here. The outer part is epidermis. This light green and gray part is cortex. Cortex is very smaller part in dicot stem. Stele is very large. Stele is the central column. It occupies the major part of the stem. Stele is the central column, occupies major part of the stem. So let us see. So this is epidermis, this is cortex. In cortex, you can find 3 zones or 3 parts. The outer cortex is also called as hypodermis. Hypodermis. This is hypodermis. Hypodermis. This is hypodermis. It is generally 2 to 3 layered cells are seen. In dicot stem, in most of the dicot stems, the hypodermis, that is outer cortex, is made up of parenchyma. Hypodermis in most of the dicot stems is made up of parenchyma. Parenchyma, as already in tissues, plant tissues, we were discussing. Ground tissue, it is a type of ground tissue. It provides mechanical support. It is a living tissue. Sometimes it may contain chloroplast. Hence, it can also synthesize food materials. It helps in photosynthesis also. In addition to providing mechanical support, it can also prepare food materials. It can also prepare food materials. This is the part. Generally, in sometimes like cucurbits, you can find chloroplast in hypodermis. Hypodermis is the external cortex or outer cortex. The outer part of the cortex is called hypodermis. Why it is called hypodermis? Dermis means epidermis, outer layer. Below the epidermis, you can find the first part of cortex. Hence, it is the name named as hypodermis. Next part, middle. This green color part is there now. That is middle cortex. That is middle cortex. It is few layered. It is made up of few layered cells. Generally, it is parenchyma. And sometimes chlorenchyma tissue is also maybe there. Chlorenchyma tissue may be also there. But inner side of the middle cortex, you can find parenchyma. It stores food materials. Parenchyma stores food materials. And the innermost layer, this is light orange color layer, you could see, that is called endodermis. Endodermis is the innermost layer of Endodermis is the innermost layer of cortex. Innermost layer of cortex. Cells are barrel shaped, closely packed without intercellular spaces. Generally, in middle cortex, you can see the intercellular spaces, but in the innermost layer of the cortex, that is called as endodermis, is lacking intercellular spaces. Generally, in dicots, endodermis or endodermal cells store starch grains. Hence, it is also called as starch sheath. Endodermis is also called as starch sheath. I'll repeat once again. Cortex is the second part of primary dicot stem. It lies between epidermis and stele. It is differentiated into three parts or three zones. The first one is hypodermis. Hypodermis is also called as outer cortex. It is generally made up of, in most of the plants, parenchyma. Parenchyma is a mechanical living tissue. Sometimes in cucurbits, it may contain chloroplasts. That's why the young primary stems are green in color. In addition to providing mechanical support, it can also prepare food materials by the process called photosynthesis. And middle cortex. Middle cortex is made up of simple parenchymatous tissue. Sometimes in some plants, part of chlorenchyma may be also seen. Chlorenchyma is also maybe seen in some plants. That is in rare cases. And sometimes you can notice this yellow color spherical structure could be seen in the enlarged portion, sector of the dicot stem. That is nothing but, that is mucilaginous canal. In some plants, this is not common in all the dicot plants, but in some plants, the mucilaginous materials may be secreted by stem. That mucilaginous material will be present in the mucilaginous canals. This is mucilaginous canal. This mucilaginous canal is located in the middle cortex. Middle cortex. Then the last part of cortex is endodermis. This is the innermost layer of the cortex. Generally, the other name of endodermis in dicot, primary dicot stem, it is called as starch sheath, since it encloses starch grains. Many starch grains will be present in the endodermal cells. The cells are barrel-shaped, closely packed without intercellular spaces. This is the last layer. It is like a sheath around the stele. That's why it is called starch sheath, since it contains starch grains. This is about cortex. Next, let us see the stele. This is the third part. Third part. This is the largest central column. This is the largest central column. Let us see once again. This is stele. The large part is there now. The central large portion is occupied by stele. Here, this dark color patch-like structures could be seen in the internal structure. This is the ground plan. You can see these are vascular bundles. These are vascular bundles. And this is medulla or pith. Medulla or pith. And the tissue present between these vascular bundles, this radially extended part of the medulla is called medullary ray. These are vascular bundles. This is medulla or pith. These are medullary rays. And the outer part of stele is called pericycle. Pericycle. This is the largest part. Let us see this structure in detail. In detail. So, stele is differentiated into three parts. The first part of stele is pericycle. The second part is vascular bundles, as we have seen earlier in the screen. The image was shown. They are vascular bundles. Now you can see the enlarged part very clearly. This entire structure is called vascular bundle. This is vascular bundle. And the next one is this pith. Pith is also called as medulla. Pith is also called as medulla. This tissue is there, light blue color tissue could be seen between the two adjacent vascular bundles. The medulla is extended up to pericycle. That is called medullary ray. Medullary ray. Since it is a primary stem, this ray is also called as primary medullary ray. Primary medullary ray. Next, let us discuss each part in detail. The first part of stele is pericycle. Peri means around. Peri means around. Cycle means the circular spherical central column is called stele. This is the outermost part of the stele. It encloses the entire stele. That's why the name is pericycle. That's why the name is pericycle. Peri means around. Cycle means around. Around the stele, you can see the pericycle. Pericycle generally in dicot stem, in most of the dicot stem, particularly in sunflower, it is made up of sclerenchyma. As you know, I think you know the types of ground tissues: parenchyma, collenchyma, sclerenchyma. All the three tissues could be seen in the dicot stem. General cortex is made up of parenchyma. Hypodermis is made up of collenchyma. Pericycle is made up of sclerenchyma. In dicot stems, generally, the pericycle may be like patches just above the vascular bundle as a cap, but in sometimes it may be like a continuous ring. It is like a continuous ring, or it is like a patch over the vascular bundle as a cap. As a cap. This is pericycle. Pericycle is made up of sclerenchyma. Sclerenchyma is a dead tissue. Sclerenchyma is a dead tissue. It provides mechanical support to the vascular bundle. Vascular bundle. This band-like structure appears like a cap. That's why it is called bundle cap. Bundle cap is nothing but it is a part of pericycle. It's a part of pericycle. Okay. Next one, vascular bundles. So this is the overview. Before that, let me explain the vascular bundle. This is the vascular bundle. So forget about this monocut vascular bundle. So this is the second right side image you can observe. This is the right side image. This is the dicot vascular bundle. Dicot vascular bundle. So you know, this vascular bundle is collateral vascular bundle. Why it is called as collateral vascular bundle? Since xylem and phloem are the same radius, one above the other. Innermost vascular tissue in the vascular bundle is xylem. The vascular tissue which is present just periphery to the pericycle is phloem. Phloem is towards the periphery. Xylem is towards the center of the stem. Xylem and phloem lie on the same radius. That's why they are side by side. That's why they are called as collateral vascular bundles. I'll repeat once again. Xylem and phloem are together side by side on the same radius. Hence, it is called collateral vascular bundle. In dicot stem, vascular bundles are collateral. They are few in number. You can count 20, 15, 16, 18. The number is definite. The number of vascular bundles in dicot stem are definite. Particular number could be seen. You can count the number. Limited number of vascular bundles. They are arranged in a circle. In the previous image already you have seen. In previous image, I'll show you once again. So this circle, each one is vascular bundle. The vascular bundles are collateral because this light blue color and red color tissues could be seen. They are on the same radius. That's why these vascular bundles are called collateral vascular bundles. And as you see, these collateral vascular bundles are limited in number, arranged in a circle. This type of steel is called U steel. U means true. E-U-S-T-E-L-E, U steel. U steel is true steel. What is U steel? U steel is a type of steel in which the limited number of vascular bundles are arranged in a circle. U steel is a true steel with a limited number of vascular bundles which are arranged in a circle. That is called U steel. And each vascular bundle, as I told you, each vascular bundle is collateral and open type. Let me show you detailed structure of vascular bundle. Under the microscope, you can find such structure. If you enlarge the vascular bundle under the microscope, you can see this type of tissues. Xylem, phloem together on the same radius, collateral, and open type. There is a strip you can see between the xylem and phloem. The pale color strip could be seen on the slide. This is called cambium. A strip of cambium is present between xylem and phloem. A strip of cambium is present between xylem and phloem. Between xylem and phloem, a strip of cambium is present. That's why this type of collateral vascular bundle is called open type. Conjoint. Why they are called conjoint? They are together. Collateral. Why they are called collateral? They are xylem and phloem are side by side. Why they are called open? Since there is a patch separating xylem and phloem. Patch of cambium could be seen between xylem and phloem. This cambium is called vascular cambium. I'll repeat. In dicot stem, the steel is U steel. The vascular bundles are conjoint, collateral, open. I'll repeat once again, 3 terms. You need to understand what is conjoint. Conjoint. Conjoint means they are together. They are together. Xylem and phloem are together. As you see, in roots, they are on separate radius. Xylem on one radius, phloem on the other radius. Here, in stem, you can see on the same radius, they are side by side. They are together. That's why conjoint. Conjoint, collateral, side by side. On the same radius, side by side. Conjoint, collateral, open. Since there is a patch of vascular cambium present between xylem and phloem. That means this patch of cambium is separating xylem tissue and phloem tissue in dicot vascular bundles. That's why they are open type of vascular bundles. Open type of vascular bundles are there in primary dicot stem. All the vascular bundles are arranged in a circle, as we have seen in the ground plan. That type of arrangement is called U steel. U means true. This type of steel is called true steel. What do you mean by true steel? A steel with limited number of vascular bundles which are arranged in a circle. The limited number of vascular bundles which are arranged in a circle is called U steel. U steel means true steel. Such type of vascular bundles could be seen in a circle. Such vascular bundles are seen in a circle in primary dicot stem. I'll repeat once again. This is very, very important. Xylem and phloem together. That's why conjoint. They are side by side in the same radius. Hence, collateral. And a patch of KMBM is present between xylem and phloem. Hence, it is open. Three different terms: conjoint, collateral, open type of vascular bundles are there in dicot stem. And they are limited in number. Their number is limited. You can count 15, 16, 18, 20, 25, 22, like that. You can count. There is a limited number of vascular bundles are seen. That's why they are arranged in a circle. They are arranged in a circle just below the pericycle. That's why this stele is called U stele. Let me check this. We'll go to the next slide. See. So this is epidermis. This is cortex. The entire circular portion is called stele. It is like a central column. It occupies the large part of secondary, primary stem. Large part of dicot stem is occupied by central column. So this is stele. And the central portion is called the empty, which is made up of simple parenchyma, that is called pith or medulla. Pith or medulla. This is simple parenchymatous tissue. It is a ground tissue. Sometimes in some plants, this is hollow. Because of the disintegration of pith, there is a hollow space could be seen. Like in water plants, you can see the pith is hollow. Hollow pith could be seen. A central empty space could be seen. Since the water plants need to be float in the water, they should be light in weight. They have to maintain buoyance. They need to float on the surface of the water body. That's why the pith is disintegrated, forming a cavity. That cavity is called pith cavity or medullary cavity. Pith cavity or medullary cavity. Let us see the overview. This is the cross section. Cross section under the microscope, you can find. Under the compound microscope, you can see such structure. This is epidermis. Epidermis. This is cortex. Cortex is collenchyma, parenchyma, and endodermis. And the central circular column is called stele. You can find there is a limited number of vascular bundles. The red color patches are nothing but they are xylem tissues. Xylem is always towards the inner side. Phloem is outer side. Phloem is outer side in the vascular bundle. They are on the same radius. They are side by side. They are on the same radius. They are side by side. That's why they are called conjoint, collateral, and open. There is a strip. You can see a white color ring-like structure could be seen. That is called KMBM ring. At the time of secondary growth, this KMBM ring is formed. Between xylem and phloem, there is a strip of KMBM could be seen. That's why this vascular bundle is called open type of vascular bundle. Open type of bundle. Let me conclude. How can you identify the dicot stem? Because it can be easily identified by the following characters. One important thing I missed, that trichomes. Trichomes are nothing but their epidermal hairs. You can see in enlarged portion also, you can see these are the epidermal hairs. The epidermal hairs are very thin, delicate. In some plants, they are spinous. In some plants, they are very hard. They protect the plant body. They protect the plant body. And they prevent the loss of water. That is called transpiration. They check the transpiration. Epidermal hairs check the transpiration along with cuticle. What is the function of cuticle and epidermal hairs? Epidermal hairs in dicot stem are also called as trichomes. Trichomes are nothing but they are epidermal hairs, outgrowths of the epidermis. They protect stems and they check the transpiration. They check the transpiration. Cuticle along with the epidermal hairs check the transpiration. These are the trichomes. This is epidermis. Next, epidermis is covered with thin cuticle and few stomata. What is the first character? What is the first feature of identification feature of dicot stem? Presence of trichomes, presence of epidermal hairs. The second one is epidermis is covered, the outer surface of the epidermis is covered with thin cuticle. Cuticle is a layer made up of cutin. Cutin is nothing but it is a wax-like substance. Cutin is nothing but it is a wax-like substance. It gives protection. It prevents the loss of water. It prevents the loss of water from the plant body. That means it checks transpiration. You know what is transpiration? Loss of water in the form of vapor from the aerial parts of the plant body is called transpiration. Transpiration can be checked by cuticle layer. That means cuticle layer protects stem. And few stomata are also there. Because of the presence of few stomata, epidermis also helps in exchanges of gases and also helps in transpiration and helps in respiration. This is about epidermis. And hypodermis generally made up of cholenchymatous tissue. As we were discussing earlier, the hypodermis is outermost layer of the cortex. It is few-celled structure. It lies just below the epidermis. It is made up of cholenchyma. It is made up of cholenchyma. Cholenchyma is a living mechanical tissue. It is a living mechanical tissue. And general cortex is very common, and in some plants you can find mucilage canals. You can find mucilage canals in cortex. And one more important point in dicot stem is important feature of the dicot stem, exclusively seen only in dicot stem with Casparian strips, endodermis with Casparian strips. In roots also we could notice endodermis is the innermost layer of cortex. Endodermis is the innermost layer of cortex. It is made up of single layer of cells. Its radial walls are very thick because the deposition of cell wall materials like chitin, suberin, the radial walls or tangential walls are thick because of the thickness. That thicknesses were for the first time noticed by Caspari. That's why these strips are named after the scientists who discovered them. Casparian strips. The endodermis can be recognized by one more feature in dicot stem. Large amount of starch grains will be also present in endodermal cells. That's why this endodermis in dicot stem is also called as starch sheath. Starch sheath. Intercellular spaces are absent in endodermis. And it is like a sheath around the steel. A sheath around the steel. Next, the important thing, identification feature of the dicot stem is U stele. Presence of U stele is a very important key feature of the dicot stem. U means true. U means true. Stele is a central column. Stele is a central column which occupies major part of the dicot stem. Why it is called U stele? U stele is a stele which contains a limited number of vascular bundles. Which are arranged in a circle, which are arranged in a ring. Such type of stele is called U stele. And each vascular bundle is conjoint, collateral, and open type of vascular bundles are there in primary dicot stem. I'll repeat once again. U stele is a true stele. There is a limited number of vascular bundles which are arranged in a circle. Such type of stele is called U stele. And in stele, each vascular bundle is conjoint, They are together. Collateral, they are side by side on the same radius. And they are open type of vascular bundles. Since there is a strip of vascular cambium present between xylem and phloem. In xylem, I forgot to tell you, in xylem, see, there is a series of xylem vessels could be seen, which are dark pink color structures. These are xylem vessels. The xylem vessels are uneven in size. The innermost xylem vessels are narrower, and the outer xylem vessels are larger. Outer side xylem vessels are larger, and inner side you can see the narrow xylem vessels. This narrow xylem vessels are called protoxylem, or this is metaxylem. First formed xylem is called protoxylem, later formed xylem is called metaxylem. In primary dicot stem, xylem is always, protoxylem is always towards the pith, towards the inner side of the stem. This condition is termed as endark. As we have seen, exarch xylem in roots. In roots, first formed xylem is towards the periphery, and later formed xylem, that is metaxylem, is at the center. Here in this case, reverse. Protoxylem is towards center, metaxylem is towards periphery. This is the entire structure is called xylem bundle. This is xylem bundle. These yellow color cells are seen in the slide. This is cambium, vascular cambium. It helps in secondary growth. It helps in the formation of secondary vascular tissues. That is, secondary xylem and secondary phloem are produced from vascular cambium during secondary growth. So this tissue is phloem. This is entire structure is called vascular bundle. Conjoint, collateral, open type of vascular bundles, and the endark condition is seen. Endark. Protoxylem is towards the center. In root, it is opposite. Exarch. Exarch condition is seen in roots. Endark condition is seen in stem. See. So key point, presence of endark xylem. Presence of endark xylem is the key feature of dicot stem. And last one, so medulla or pith, a large parenchyma tissue is located at the center of the vascular bundle. Stele. The central portion of the stele is called medulla or pith. Medulla or pith. That is called medulla or parenchymatous medulla. It stores food materials and it is having intercellular spaces. In some plants like water plants, medulla is disintegrated, forming a cavity, medullary cavity or pith cavity. And the tissue which is extended between the, this is medulla, this is the tissue which is extended between the vascular bundles. Between vascular bundles, there is a tissue which is extended from pith to endodermis, is called primary medullary ray. Primary medullary ray. So this is about dicot stem. Let me conclude. So dicot stem shows three parts: epidermis, cortex, stele. I'll repeat once again. The first part of dicot stem is epidermis. Second part is cortex. And the third last part is stele. Epidermis. Epidermis is the outermost layer of the stem. It is made up of single layer of cells. Cells are barrel shaped, which are closely packed without intercellular spaces. Generally, cells are colorless, But in some plants, epidermal cells may be green, but that is very rare. The outer surface of epidermis is enclosed, is covered with cuticle. Cuticle is a layer made up of cutin. On epidermis, you can find epidermal hairs. These epidermal hairs are also called as trichomes. They are multicellular structures. What is the function of epidermis? Epidermis gives protection to the inner parts. Epidermis gives protection to the inner parts. The primary function of epidermis is providing protection. Since its outer surface is enclosed, covered with cuticle and epidermal hairs, it checks transpiration. It checks transpiration. When the stem is very young, the epidermis also may show few stomata. Stomata are minute microscopic aerating pores. They help in exchanges of gases as well as they help in transpiration. Stomata are minute microscopic aerating pores. They are few in number in the epidermis. They help in exchanges of gases and also transpiration. Next, second part. Second part is cortex. This is the middle zone or middle part of the primary dicot stem. It is differentiated into three parts: the outer cortex, middle cortex, inner cortex. Outer cortex is called hypodermis. Hypo means below, dermis means epidermis. This is the outer part of cortex. It is made up of few layered of cells. Generally, the cells are parenchymatous. Rarely they may contain chloroplast. Parenchyma, that is hypodermis, provides mechanical support. Since parenchyma is a living mechanical tissue, in some plants like cucurbits, hypodermis may also contain chlorenchyma. Green parenchyma is called chlorenchyma. Hence, it helps in photosynthesis. And the second part of cortex is called middle cortex. It is made up of few layered parenchymatous cells. Cells are loosely packed with small intercellular spaces. It stores food materials. In some plants, it may contain mucilaginous cavities or mucilaginous canals. This yellow color part which could be seen in the cortex is mucilaginous canal. Next, last part is endodermis. This is the innermost layer of cortex. Innermost layer of cortex is called endodermis. Endodermis is made up of single layer of cells. Single layer of cell. Cells are barrel shaped. No intercellular spaces. They are closely packed or compactly arranged. They have one prominent feature. Their radial walls are thick. These thickenings are called Casparian thickenings. Casparian thickenings. That is the identification point of endodermis. In primary dicot stems, endodermis stores large number of starch grains. Since the cells contain large number of starch grains, the endodermis is also called as starch sheath. This is starch sheath. Next, stele. The central large column-like structure which occupies a major part of the stem. It is differentiated into three parts. They are pericycle, vascular bundles, and medulla medullary rays. Pericycle. Pericycle is the outermost layer of stele. Outermost layer of stele. It is made up of few layered sclerenchymatous cells. Few layered sclerenchymatous cells. Generally, it is like cap over the vascular bundles. It is like a discontinuous ring or continuous ring in some plants. It is like a continuous ring. Here you can see a discontinuous ring. It is like a cap just over the vascular bundle, which provides mechanical support. It provides mechanical support. A cap of sclerenchymatous tissue is present over each vascular bundle as a cap. Hence, it is called as bundle cap. It is made up of sclerenchyma. Sclerenchyma is a dead tissue. It provides mechanical support. Next one, vascular bundles. Vascular bundles are already you have seen. Let me share. You see the right side image. Vascular bundles are limited in number. Vascular bundles are limited in number. Vascular bundles are limited in number, arranged in a circle. Arranged in a circle. Such type of stele is called U stele. U stele is a true stele. What do you mean by U stele? U stele is a type of stele with limited number of vascular bundles that are arranged in a circle. That are arranged in a circle. Definite circle. They are arranged in a definite circle. Such stele is called U stele. So stele is called U stele. Next, let us see the vascular bundle detailed structure. This is conjoint, collateral, open type of vascular bundle. In each vascular bundle, xylem tissue is at the center, towards the center. Phloem is towards the pericycle. This is phloem. This is phloem tissue, which is towards the pericycle. This is pericycle patch. Cleaving gammatus, pericycle patch. Conjoint, collateral, open type of vascular bundles are there. In xylem, protoxylem is towards the center and metaxylem is towards the periphery. This condition is known as endark. This condition is known as endark. Presence of protoxylem towards center is called as, is termed as endark. This condition is also known as endark condition. Sorry, Andy. Sorry. నేను నా ల్యాప్టాప్ స్విచ్ ఆఫ్ అయింది. Sorry, let me continue. I am concluding. We are at conclusion stage. కంటిన్యూ చేద్దాం. వినిపిస్తుందా? ఓకే. మనం ఇంతకుముందు చూసాము. షేరింగ్ ఆప్షన్ పోయింది రానా? వాస్కులర్ బండిల్స్ ని కాంజాయింట్, కొలాటరల్, ఓపెన్ టైప్ ఆఫ్ వాస్కులర్ బండిల్స్ అంటారు. మనం ఇంతకుముందు ఇమేజ్ చూసాము. xylem అండ్ phloem కలిసి ఉంటాయి కాబట్టి కాంజాయింట్. ఒకే రేడియస్ మీద ఉంటాయి కాబట్టి కొలాటరల్. ఆ రెండింటి మధ్యలో కేంబియం స్ట్రిప్ ఉంటుంది, వాస్కులర్ కేంబియం ఉంటుంది, అందుకని క్లోజ్డ్, సారీ ఓపెన్, అందుకని ఓపెన్ టైప్ ఆఫ్ వాస్కులర్ బండిల్స్ అంటాం. ఓపెన్ టైప్ ఆఫ్ వాస్కులర్ బండిల్స్ అంటాం. ఈ వాస్కులర్ బండిల్స్ లిమిటెడ్ ఉంటాయి, ఒక సర్కిల్లో ఉంటాయి, యూస్టీల్ అంటాం. ఎందుకు అదే పనిగా ఇన్నిసార్లు రిపీట్ చేస్తున్నానంటే దిస్ ఇస్ వెరీ వెరీ ఇంపార్టెంట్ కీ పాయింట్, ఐడెంటిఫికేషన్ పాయింట్. హౌ కెన్ యూ ఐడెంటిఫై ది డైకాట్ స్టెమ్ హ్యావింగ్ ట్రైకోమ్స్ ఉండటం, సింగిల్ లేయర్డ్ ఎపిడోమిస్, colon-camatous clearant hypodermis. Endodermis is also known as starch sheath and eustine. And vascular bundles are conjoint, collateral, open type of vascular bundles. So compulsory in examination, you are going to get this for 8 months. You have to practice the diagram And also you need to write, you need to explain, and you need to label. A neat labeled diagram is compulsory, and description with different subheadings also compulsory for the essay. For that, you have to follow the instructions, and you need to be very careful while drawing the diagram. If you practice the diagram, you can get the you can draw the diagram in examination. Okay, let me conclude. So tomorrow we will discuss about monocot stem, internal structure of monocot stem. So I'm closing this session. Take care. Bye bye.